Angiosperms represent one of the greatest terrestrial radiations. The oldest fossils date back from the early Cretaceous, followed by a rise to ecological dominance in many habitats before the end of the Cretaceous. Darwin referred to the origin of angiosperms as abominable mystery. He was referring to the dreadful lack of fossil evidence that would help link angiosperms to gymnosperms, seed ferns, or other extant or extinct groups. The lack of fossils was attributed to the thought that angiosperms arose in dry upland areas that were not optimal for fossilization. Since Darwin's time, many advances have been made, including new fossils and lots of molecular data aimed at their mystery. Many theories and hypotheses have been proposed from time to time, and most of them have revolved around two basic theories, which include the Unential theory and Pseudanthial theory. Now, let us discuss the Unential theory first. It is also known as Anthostrobilus theory. The Unential theory was proposed by Arber and Parkins in 1907. According to this theory, the angiosperm flower is interpreted as being derived from an unbranched bisexual strobilus bearing spirally arranged ovules and pollen organs, similar to the hermaphrodite reproductive structures of some extinct Bentitian gymnosperms. The carpel is thus regarded as a modified megasporophyll. The bisexual flower of magnoliales has been considered to have evolved from such a structure. Now, let us discuss the Pseudanthial theory. Commonly associated with the Anglerian school, the theory was first proposed by Wettstein in 1907. According to this theory, wind-pollinated cone-bearing naked seed plants may be the ancestors of angiosperms. Engler and others concluded that angiosperms evolved from conifers, neophytes, and certain catkin-bearing angiosperms such as the forerunners of Casuarina equistifolia. The existing theories and hypotheses about the origin of angiosperms include Anthocorm theory. This theory is a modified version of the Pseudanthial theory and was proposed by Neumayer in 1924 and strongly advocated by Meuse. According to this theory, the angiosperm flower has several separate origins, that the angiosperms are polyphyletic in origin. In most magnolidae and their dicotyledonous derivatives, they are modified pluriaxial system that originated from the Notopsids via the piperales, whereas the modification of an originally uniaxial system gave rise to flowers of Chloranthaceae. Meuse in 1963 postulated a separate origin of monocotyledons from the fossil order Pentoxylales through the monocot order Pandanales. Now, let us discuss Anthophyte hypothesis. In 1986, Doyle and Doyle proposed that flowering plants were derived from Neophytes. The Anthophyte hypothesis was developed further in later years. However, after reconsidering incongruent molecular, phylogenetic and morphological data, the Anthophyte hypothesis was rejected by the authors in 2000. Another theory is woody magnoliid theory. Several theories propose that modern flowering plants evolved from magnolia-like species having strobiloid flowers with many whorled parts, not unlike bennettite aliens. Another theory is Paleoherb theory or Paleoherb hypothesis. According to Taylor and Hickey in 1992, flowering plants evolved from herbaceous forms possessing ovules and pollen-bearing organs that combined over time, producing modern flowers. The term Paleoherb was first used by Doyle and Doyle in 1989 for a group of derivatives of magnoliid having anomocytic stomata, two whorls of perianth, and trimerous flowers, including Lactoridaceae, Aristolochiaceae, Cabombaceae, Pipereales, Nymphaeaceae, and monocots. Another theory is Chloranthoid hypothesis. This hypothesis suggests that flowering plants evolved from chloranthoid ancestors, not unlike the modern Angiosperm family Chloranthaceae. Arguments presented by Stuessy in 2004 in favor of Leroy's proposal include reports of data from molecular systematics that place Hydeiosmum in a clade basal to other extant angiosperms. Multiple origin hypothesis. According to this hypothesis, the flowering plants evolved from multiple unrelated seed plant lineages. The Polyphyletic-Polychromic Polytopic hypothesis and Nair's Triphyletic theory are best placed in these hypotheses. Another theory is Gonophyll theory. Ronald Melville was the main proponent of the Gonophyll theory, according to which the flowering plants evolved from glossopterids of Mesozoic Gondwana. Together with Voltzelian conifers, Gigantopterids, and glossopterids are probably the best known fossil seed plant groups of arid, sparsely vegetated Pangean biomes of Permian period. Of the aforementioned plants, at least presently known, only conifers and some glossopterids persisted beyond the Permo-Triassic extinction event that occurred 251 million years ago. Another important hypothesis is seed fern hypothesis. Several differing seed fern hypotheses promulgated by American, Asian, and European paleobotanists suggest an origin of flowering plants from Paleozoic and Mesozoic gymnosperm seed fern stock. Research findings that shed light on the origin of flowering plants from pteridosperms include Doyle and Frohlich. Doyle and others suggest that the cupule forming Catonia or other as yet unstudied seed ferns were the ancestors of angiosperms. However, more work is needed to understand the anatomy of ovules and fruit bearing rachis of seed ferns or to ascertain homology with functionally equivalent features of the unripened seeds, carpels, and fruits of angiosperms. Another hypothesis is biome and paleogeographically specific hypothesis. Interestingly, both on upland hypothesis and coastal hypothesis were proposed to explain topographical centers of origin and radiation of first flowering plants. According to Stebbins, alpine biomes of northern latitudes might have been of center of early radiation of the angiosperms. A similar idea, The Eastern Asian Center hypothesis was put forth by Sun et al. In 2001. Based on the recovery and study of fossil pollen casings recovered from the deep sea drill holes, Hochuli and Feist-Burkhardt in 2004 suggested that early flowering plants might have evolved in a boreal cradle. Field et al. Based their conclusive ideas on early angiosperm ecology from the phylogenetically robust Anita clade. However, despite possibly wide agreement among certain workers that Amborulaceae and Hydatilaceae are basal angiosperms, Amborula trichopoda is almost certainly not an early flowering plant. Developmentally and genetically based theories. Ideas on the origin of angiosperms based on evidence from the study of ontogeny of seed plants include Asama's growth retardation theory, Mayon's gamoheterotrophic hypothesis, Frohlich and Parker's mostly male theory, and Becker and Thiessen's out of male and out of female hypothesis. The out of male and out of female hypotheses are based on a floral homeotic protein quartet model and evolutionary development of pollen bearing and ovule organs of cone axis. Now, unifying theories. Bona fide theories fall into this category. Stuessy's transitional combinational theory, which draws on information from several scientific disciplines is the most up to date theory in this category. Stuessy proposes that carpels evolved first, followed by double fertilization and then by flowers slowly over many millions of years, perhaps more than 100 million. Through contributions from Paleobotany, Phylogenetics, Classical Developmental Biology and Modern Developmental Genetics, tremendous progress has recently been made in elucidating the origin and diversification of the angiosperms. Fossils have contributed a lot. Paleobotany has at least 3 crucial roles to play in resolving the origin and early diversification of flowering plants and addressing the following questions, which include: what lineage is ancestral to the angiosperms? What were the first angiosperms and how have characters evolved? Fossil discoveries and analysis of existing data have made significant contributions to our understanding of the possible ancestors of flowering plants as well as to the early diversification of angiosperms. Third question is, when did the major clades of angiosperms diversify on the basis of fossil calibrations and molecular based methods? Now, let us discuss ancestors of flowering plants. The closest relatives of angiosperms remain a mystery. Phylogenetic analysis of morphological features conducted in 1980 suggest that Nettles were the closest living relatives of angiosperms. Cladistic analysis of extant and fossil taxa recovered a clade of Bintitials, Pentoxylales, Nettles and angiosperms. Doyle and Doyle in 1986 named this clade the Anthophytes in reference to the flower-like reproductive structures in all members. The Anthophyte hypothesis that angiosperms belong to this clade and are possibly sister to the Nettophytes concomitantly had a profound effect on views of the evolution of the angiosperms. Molecular phylogenetic studies, however, later revealed that Nettles are not the sister groups to the angiosperms, but the extant gymnosperms form a clade with Nettles most likely associated with conifers. The demise of the Anthophyte hypothesis profoundly altered views of angiosperm origins. Accompanying this demise is a need for alternatives. If no extant gymnosperm is sister to angiosperms, what fossil lineages, if any, are closely related to flowering plants? Doyle in 2001 and Soltis et al. In 2005 concluded a series of analyses of morphological and molecular data sets involving extant taxa as well as fossils to reassess the closest relatives of the angiosperms. These analyses yielded similar results and revealed a revised Anthophyte clade. In this clade, Catonia is sister to the angiosperms, followed by Benititials, Pentoxylon and Glossopterids. Hence, fossils such as Catonia and Benititials may be the closest relatives of angiosperms. However, the recent discovery that Neophytes and Benititials share features suggests that further attention be given to circumscribing and evaluating a revised Anthophyte clade. Although these analyses are useful, one of the biggest remaining challenges facing evolutionary biologists is ascertaining the fossil lineages that represent the closest relatives of angiosperms. Do Catonials and Benititials actually represent these lineages? Are there key fossil links that remains to be discovered? The earliest angiosperms, which are fossils, there are two highly divergent views on the general habit of the earliest angiosperms: woody and terrestrial or herbaceous and aquatic. This has actually been a long-standing debate. The hypothesis that the earliest angiosperms were woody is supported by the fact that the most basal angiosperms are woody and all gymnosperms are woody, as well as the fossil lineages that are considered most closely related to angiosperms, for example, Catonials and Benititials. Amborilla, the sister to all other living angiosperms, is woody, as are members of Astrobiales, another early branching lineage of living angiosperms. However, an aquatic origin of angiosperms is supported by the fact that several of the earliest known fossil angiosperms were aquatic. Archifractus represent perhaps the oldest, most complete angiosperm fossil. It is estimated to be approximately 115 to 125 million years old. On the basics of morphology, it clearly was aquatic. The phylogenetic placement of the fossil Archifractus as sister to all extant angiosperms plus the near basal phylogenetic position of extant nymphales lends support to the view that the aquatic habit arose early in angiosperms and that perhaps the earliest angiosperms were aquatic. However, later analyses questioned the placement of Archifractus as sister to all extant angiosperms. Some analyses of Frisch et al. Placed Archifractus with water lilies. The recent discovery that Hydatilaceae are part of water lily clade and hence a new branch near the base of the angiosperms greatly increases the morphological diversity encompassed by the nymphales clade. This finding raised the possibility that nymphales were once much more diverse and could have encompassed Archifractus as well as other now extinct lineages. Hence, it now seems prudent to keep an open mind regarding the placement of Archifractus. It represents well the difficulty in placing fossil lineages that are morphologically distinct with no clear synomorphies with extant taxa. Another early angiosperm fossil was considered a possible water lily relative by Frisch in 2001. This fossil is debated at approximately 125 to 115 million years old and was used as evidence to support the antiquity of the Nymphaeals lineage. This putative ancient water lily fossil is therefore extremely important in discussions of the diversification of Nymphaeals as well as the angiosperms. A phylogenetic analysis using the morphological data set of Les et al. In 1999 for extant Nymphaeals placed the fossil as sister to Nymphaeals. Synapomorphies with extant Nymphaeals include a syncarpous gynoecium, a perigynous or epigynous perianth, and a central protrusion of the floral axis between the carpels. However, neither of the last two characters is found consistently throughout Nymphaeals. The unnamed fossil exhibits features shared by both Nymphaeals and Elysium, which belongs to Austrobiales and occurs in a site with abundant fossil seeds attributed to Elysiates. The recent floral analysis and dating experiment of Yeo et al. In 2005 raised the possibility that the unnamed fossil of Frisch et al. May have actually been part of an ancient assemblage that included Nymphaeals and Austrobiales. One of the most exciting recent developments in the study of angiosperm evolution has been the enormous progress made in elucidating angiosperm evolutionary history. Figure depicts a summary tree of origin of angiosperms and angiosperm relationships. Phylogenetic analysis based on an ever increasing number of gene sequences have clearly and unequivocally established the first branches of extant angiosperm diversity. Many studies have revealed strong support for the sister relationships of Amboralaceae, followed by Nymphaeals and then Austrobiales to all other extant angiosperms. In most analyses, the monophyletic Amborella is followed by water lilies or Nymphaeals and Cabomberaceae and Hydatylaceae. Although a few investigations placed Amboralaceae plus Nymphaeals as sister to all other angiosperms. Recent analyses involving complete or nearly complete plastid genome sequences have placed Amborella alone as sister to all other extant angiosperms. Although this now appears to be the favored topology, it should be confirmed using nuclear sequence data. An exciting recent molecular finding is the placement of Hydatyla in the Nymphaeals. Hydatylaceae had been placed in the monocots, but molecular data revealed in a state that the family is part of the ever early diverging angiosperm clade Nymphaeals with high support values. Hence, this is a dramatic phylogenetic shift. This result is important in that it greatly expands the morphological diversity encompassed by Nymphaeals. For example, Rudall et al. In 2007 proposed that the most plausible interpretation of the reproductive units is that each such unit represents an aggregation of reduced unisexual apetalous flowers. This type is different from flowers of Nymphaeals. However, Hydatyla has ascidiate carpel development consistent with placement in Nymphaeals. The placement of Hydatyla within Nymphaeals raises the possibility that this clade was once far more diverse morphologically and increases the plausibility that the morphology of Archifractus could be encompassed by this clade as originally proposed by Friss et al. In 2003. After Amborella and Nymphaeals, an Ostrobileales clade of Iliaceae, Ostrobileaceae and Trimeniaceae is the subsequent sister group of all remaining Angiosperms. Whereas these basal most branches are well supported and resolved, relationships among the remaining major lineages of Angiosperms have been unclear and difficult to resolve, with 3, 5, 9 and 11 gene data sets. These remaining Angiosperms or Mesangiosperms are magnolids, Chloranthaceae, monocots, Ceratophyllaceae and eudicots. Analyses of multi-gene data sets with adequate taxon sampling have provided strong support for monophyly of each of these clades of Angiosperms. However, different analyses have depicted various relationships among these 5 clades with only weak support. In contrast to early studies, recent analyses based on complete or nearly complete plastid genome sequences appear to resolve relationships among these 5 lineages of Mesangiospermi. After the grade of Amborella, Nymphaeals and Ostrobileales, Chloranthaceae and magnolids may form a clade that is sister to a large clade of monocots and Ceratophyllaceae plus eudicots. The clade of monocots as sister to the eudicots plus Ceratophyllum clade is strongly supported. Jensen et al. In 2007 also found that these relationships, except that their initial analyses did not include Ceratophyllum. The first 3 living branches were relatively easy to recover in phylogenetic analyses, a result reported in some of the initial multigene analyses of Angiosperms. These results suggested a more gradual diversification process or greater extinction at base or of the Angiosperm tree. However, after some initial early experiments in Angiospermi, the Angiosperms rapidly radiated, yielding the 5 lineages of Mesangiospermi. This is an exciting example of an important molecular insight into Darwin's abominable mystery. The fossil record certainly supports the presence of many diverse lineages early in Angiosperm evolution, but phylogenetic analysis of extant Angiosperms indicate that the radiation responsible for nearly all extant Angiosperm diversity was not associated with the origin of Angiosperms, but occurred after the diversification of Amborella, Nymphaeals and Ostrobileales. A well-resolved phylogeny for extant Angiosperms has been used as a framework in an effort to reconstruct the habit as well as other morphological features of the earliest Angiosperm plants. As noted, Amborella, which is sister to all other extant Angiosperms, is woody, as are Ostrobileales. Anatomical and physiological features of Amborella, which lacks vessel elements, and Ostrobileales, which have primitive vessel elements, considered intermediate between tracheids and vessel elements, prompted the hypothesis that the earliest Angiosperms were understory shrubs, perhaps without vessel elements and with lower transpiration and stem water movement than in typical of most extant Angiosperms. Such plants occupied habitats referred to as dark and disturbed and prompted the hypothesis that the first Angiosperms were understory shrubs living in partially shaded habitats that depended on canopy disturbance to open up new sites for colonization. Outgroup comparisons favor a woody habit for the first angiosperms. Dating origin of the angiosperms. In an attempt to estimate the timing of angiosperm origins, various authors have used a variety of different molecular data sets and estimation procedures. Although the oldest angiosperm fossils date to 132 million years, molecular estimates have been somewhat older. In recent years, studies estimating the age of crown group angiosperms appear to be converging on estimates of between 140 and 180 million years. This apparent convergence in age estimation is probably due to the increased number of characters used as well as the use of new relaxed clock method that allows rates to vary across the tree. Also, recent studies have used multiple fossils as either calibrations or maximum and minimum age constraints. Extant members of old lineages represent recent diversifications. The divergence time estimates of Yu et al. 2005 suggest that the crown group Nymphaeals date to the Eocene. Nymphaeaceae and Cabombaceae split at that point. Extant genera of Nymphaeaceae began to diversify in the late Eocene to early Oligocene, and the two extant genera of Cabombaceae diverged during the Miocene. These results indicate that extant Nymphaeals diversified relatively recently, whereas the stem lineage to Nymphaeals is old on the basis of fossil attributes to Nymphaeals from early Cretaceous. Although the Friss et al. Fossil attributed to Nymphaeaceae from the middle Cretaceous may be best placed deeper in the angiosperms, there is a distinct gap between the origin of Nymphaeals and its diversification into modern lineage. It is important to note that diversification occurred in a narrow window. Many of the divergence of major clades of angiosperms occurred rapidly. For example, molecular dating techniques provide a time frame for the likely rapid diversification of 5 major lineages of Mesangiospermi, magnolids, monocots, Chloranthaceae, eudicots, and Ceratophyllaceae. This diversification ultimately yielded perhaps 97% of all angiosperm species was rapid, occurring over a span perhaps no more than 5 million years. In perspective, this span represents a time frame comparable to the rapid radiation of the Hawaiian silverwort alliance, which putatively arose from North American ancestors 5 million years ago. Subsequent radiations within the clades of Mesangiospermi also occurred rapidly. For example, the Saxifragales and Rosid clades also diversified over similar and narrow time spans. The rapid diversification within the Rosid may be of particular importance in that the inferred bursts of diversification correspond in timing with the rapid rise of angiosperm dominated forests as suggested by the fossil record. The Rosid diversification also corresponds to the diversification of several other lineages that apparently evolved in parallel with the diversification of angiosperm forests. A similar window of diversification is seen in the tree species in clades over than the Rosids. Cornels of the asterid clade also appears to have diversified during the same window of time as did the woody members of Saxifragales.